Hello everyone, welcome to CRL webinar series. I'm Dr. Kai Leng, the Regional Field Application Scientist of Azure Biosystems. Today, I will talk about the technical advancements in imaging and scanning technologies for high-impact research. I'll be using Sapphire Biomolecular Imager to explain what are the most efficient and advanced technologies to do RGB and near-infrared fluorescence imaging, chemiluminescence imaging, and phosphor imaging on biological samples. I will also give you a few tips on how to do accurate quantitation in Western blotting, especially about fluorescence Western blotting. First, let me introduce Azure Biosystems to you. Azure Biosystems is founded based on innovation. It's co-founded by Arnold Shifji and Dr. Diping Chair. Arnold is our CEO and he's a venture investor and entrepreneur, while Dr. Diping Chair is our executive VP of R&D. He led the development of Illumina expression array imager and then developed imaging images while he was at Alpha Innotech. Alpha Innotech is a brand that we all are quite familiar if you're doing Western blot. And he also one of the first to develop a quantitative fluorescence Western blot 3D imager while he was a VP of R&D at Alpha Innotech. Azure Biosystems founded in 2013. We based in Dublin, California in the United States, very near the Silicon Valley. There we designed, develop, and manufacture all our products. The core members of Azure Biosystems, including Dr. Diping Chair, are come from Alpha Innotech, which then acquired by Cell Biosciences in 2009, then Protein Simple in 2011. Hence, the birth of Azure Biosystems. We as a company, we offer a full range of leading products to the market. We have been specializing in protein expression imaging since 2014. This is our first CCD imager that use both laser and LED excitation light sources for quantitative fluorescent and chemiluminescence Western blot. In 2013, we launched the industry first hybrid CCD and laser scanning system, which called Sapphire Biomolecular Imager, which is the example that I will be using today to explain about advanced technologies. In 2018, we have reagents to couple with and complement with our Western blot workflow. And 2019, we developed the upgraded version of Azure imaging systems, revamped the software to design and meet the rigorous publication standards while still being very flexible. And last year, 2020, we launched Azure Cielo real-time PCR systems to venture into the genomic market. Okay, Sapphire Biomolecular Imager. I'll be using this imager to take a deep dive into imaging technologies. So why? Because it is the first and only laser scanner that can capable of doing all these detection. First is RGB, red, green, blue, visible fluorescence laser detection, NIR fluorescence laser detection, CCD-based chemiluminescence, and also phosphor imaging. This scanner uses 4 solid-state diode laser excitation light sources. We have 4 channels here, 488, 520, 685, and 784 nanometer wavelength. This is a table of compatible dyes that can be used for these 4 excitation light sources. In terms of excitation light sources, not all near-infrared signals are the same. So here I want to limit the context into fluorescent excitation light sources, particularly about near-infrared signals. I put up these 2 light sources, LED, light emitting diode, and laser diode, to explain to you what are the differences in terms of excitation light sources. Let us look at LED light source. It is a broadband light source, giving you relatively low power output with diffused light and a small compact source. And of course, it comes with economical price. You can get great data from it, but you do still get a little more background that you might not want, which is why laser diode is used in this scanner for those who prefer the fluorescence imaging at a much higher, I'll say, performance rate. Laser diode. This is a monochromatic light source, has much higher power output. It is collimated light to deliver more energy to the sample than deliver premium performance as well. Here in this figure, the excitation is right at the peak of the spectra right here, which means very little to no crossover or leakage over the emission wavelength. Gives you the black and clean background every single time because there's a less light leakage due to the narrow excitation and excite close to the excitation peaks, which means you have higher emission signal and best signal to noise. Now I'm going to the detection technologies that we use in the system. First up, photon multiplier tubes, aka PMT. It is an electro-optic device converting light energy into electrical current and amplifying the current on the way. So it is highly sensitive due to this internal gain that can amplify signals. Second, avalanche photodiode, APD. This is a highly sensitive semiconductor electronic device that exploits the photoelectric effect and convert light into electricity. So from a functional point of view, they can be regarded, both of them, PMT and APD, can be regarded as the semiconductor analog of photon multiplier, either by the vacuum tube in PMT or in the manner of snowball effect in the APD. Next is charge coupled device, CCD, which is an integrated circuit etched on a silicon surface, and photo incidence on this surface will generate charge that can read by electronics and then convert it into a digital copy of the light patterns that falling on the CCD chips. But not all CCDs are the same. Let's take a deeper look into it. So this is the charge coupled device, CCD. As we all know, it can capture the entire field of view simultaneously, and we as a user can control the integration time. You can choose to have a long or short exposure time depending on your application. It is ideal to image luminescence and white light imaging, especially those signals that varies across time. In this case will be chemiluminescence. It gives you high sensitivity in chemiluminescence imaging with long exposure time, for example, up to 60 minutes exposure. And as I said before, CCD allows you to do simultaneous light detection in a big area. First, it allows you to do quick imaging of DNA gel and stained protein gel, and also 3D objects such as agar plate and small animals and so on. However, CCD technology is limited in terms of it has low quantum efficiency in long wavelength, those up to 490, 500 and above. It is better than film, of course, but it has also limited dynamic range. Now this brings us to dynamic range of a CCD. So what does it really mean? And is there any difference between the CCD chips or not? And it clearly is. The size of a CCD means how much electrons can actually hold in a pixel well. So dynamic range is defined as the full well capacity divided by the camera noise and related to the ability of a camera to record simultaneously very low light signals together with bright signals. What is full well capacity? Full well capacity is the maximum number of electrons that register a signal in a pixel. So this is a pixel. Larger pixel, in this case, look at indicated by second CCD chips, will have higher well capacity, which also lead to higher sensitivity, better signal to noise ratio, and increased dynamic range. You can see here, this is so for CCD chip 1, you have pixel well capped at 25,000 electron, and then for CCD chips 2, which we use in the Sapphire biomolecular imager, it capped at 40,000 electron. Okay, so when you image a sample, a blood, for example, having 32,000 electron, it will be saturated in CCD1. So what it means is saturation, when the electrons cannot be filled anymore into this pixel well, the charge is exceeded and reach the saturation level or exceed it, the camera will start to deviate from the linear response and then compromises the quantitative performance of the camera. And you see that when you have a larger pixel well cap, the pixel can actually capture more light, or in this case, electron, before reaching saturation. Thus, larger well capacity means the pixels can capture more light before saturation, thus greater dynamic range. So back to the example that we use to understand advanced technologies. So in terms of laser scanning technology, we know that scanner uses scan head containing a point source laser diode coupled with the PMT or APD detector. The scan head will move back and forth across a defined field of view, exciting the fluorophores and detecting the emission signals simultaneously. In terms of laser scanner, it is best to image high sensitivity fluorescence imaging, quantitative Western blot, especially when you need it to be cover a wide dynamic range. And it is also ideal if you want to look for high resolution scanning at 10 micro resolution for on samples such as tissue sections, arrays, and so on, including cell culture plate imaging. But laser scanners are also limited. They can do 3D object imaging, but of course, it cannot be compared with, in this case, in vivo imaging systems. For laser scanner, sometimes the scanning time will take longer. And then in terms of chemical imaging, there are limited control of exposure time as well. Laser scanner detector, in this case, we're talking about fluorescence, RGB, and near infrared fluorescence imaging, and also phosphor imaging. We have two detectors I mentioned before, PMT and APD. The keywords for these two technologies are quantum efficiency, QE, and the spectral response when you excite the fluorophores and then the receiver of it. PMT is highly sensitive photodetector in RGB fluorescence due to the high quantum efficiency over visible spectra. Thus, it is best for Sci2 detection and also phosphor imaging. And second, avalanche photodiode APD, it has high quantum efficiency in the near-infrared fluorescent signals at the longer wavelength, starting from 600 nanometer wavelength and above. Thus, it is best, I'll say, to detect Psi3, Psi5, and near-infrared channels. The combinations of PMT and APD is the best if you want to image fluorescent samples because the signals will not be compromised at either extremes of the spectrum or any points in between. Every single detector technology has their own intrinsic dynamic range. We want to look for wide dynamic range in this detector. So what is wide dynamic range means? It means the ability to capture both very low and very high signal intensity in the same image without saturation. Here we have X-ray film, which is very sensitive but also easy to saturate because you can see the dynamic range is giving 1 to 2 optical density. Next, we have CCD chips giving you 3 to 4 optical density. And finally, PMT and FPD gives you has the widest dynamic range between four of them. All right, so this is the example that I talk about today, Sapphire biomolecular imager. It is a combination of three different detectors, okay, PMT, FPD, and CCD, and together it uses laser excitation light sources, four of them. So it gives you the flexibility in terms of RGB visible fluorescence, near infrared fluorescence, phosphor imaging, and also chemiluminescence imaging in a single imager. It allows you to do a lot of samples in one go, thus increase and speeding up your workflow. And then, as I said here, we're talking about fluorescence, chemi, and also phosphor imaging. The samples is not longer limited to, let's say, blot or membrane. You can image gel, plate, slices, array, and animal and organs as well. Because this is a scanner. Scanner has their certain height allows you to fit the samples into it. This is a list of fluorescence application, covers from membrane, cell-based assay, tissue section, molecular imaging for such as in vivo imaging, that is a very initial or prelim, I'll say, gel and in vitro assays. Now I will talk about accurate quantitation for fluorescence Western blot since we're talking about fluorescence imaging here. So here we have a fluorescence Western blot with only infrared 680 secondary antibodies incubated, and we image it under both infrared laser and also LED light sources. You can see that both of them gives you very good signal when it's imaged under 680 channels, but you will see the differences once you image the blot under uh, infrared 800 channels. You will see LED will give you a little bit of crosstalk or say leak over at the 800 channels, while laser excitation light source will give you significantly lower fluorescence imaging on a blood, for example. And then I wanted to detect two proteins, total proteins, let's say proton proteins and phosphorylated protein. I'm using infrared 680 secondary dyes and infrared 800 dyes. This we need to be careful in terms of the imaging capability of the system, whether it will gives you this or say leakage or crosstalk, because we need to quantify these two proteins. So any crosstalk that resulted from, let's say in this case would be LED light sources, will makes it very difficult to give you accurate quantitative expression later. And here I want to highlight what is the benefit of doing fluorescence imaging, especially fluorescence Western blotting. It allows you to do multiplexing and detect multiple proteins and also including normalization states at the same time in one single blot. Thus, it improves reproducibility and better Western blot quantitation. We do not need to worry about the discrepancy if you're running two different gels of the same samples, for example, and then you don't need to worry about human errors and so on and so forth if you want to detect up to four proteins in a single blot. It is the worry where you don't need to have multiple plots and extending the time of your Western blotting workflow because you can compare bands on the same blot. And second, you can include total protein normalization in your Western blot that will significantly improve the quality control on your Western blot, no matter the samples has been tested by different, I'll say, lab tech or personnel. Right. So the power of four laser in this case, it gives you quantitation power. You can detect two proteins and normalize for housekeeping control. This will be in the case of fluorescence Western blot, and also include another channel for total protein stains. And here, this is an in-cell Western detecting two proteins and normalize a total cell stains. An instrument like this will actually speed up the biomarker identification workflow. As we know that we need multiple scientific tools and experimental design to prove a single scientific hypothesis at many levels, such as subcellular levels, in the cellular levels, tissue levels, and up to organism levels. When you include more and more control groups and the levels are increasing, the complexities is also increases and the workload and time needed will also increase significantly. That's why I will encourage that we should utilize a scanner or particular laser scanner with a good detector to speed up this workflow because it allows you to put a lot of sample and scan it in a go. So you don't need to do it repeatedly, and it allows you to do a prelim scan just to check whether your optimization is correct or not. This is very good if you have a lot of workload, you want to do high throughput Western blotting, immunohistochemistry. All right, so here comes the fun part. These are the publications we received over the years, but I only extracted those published 2020 and to today. Firstly, of course, a laser scanner with a CCD detector allows you to do high throughput chemiluminescence imaging. You can see this is the publication where they do Western blots on the samples collected for cystic fibrosis patients, where they tested against different treatment and their markers. And second, this is a publication published in Basic Research in Cardiology. It is around 11 impact factor. Of course, they did a lot of research, not only on Western blot, but here I want to say that they use this laser scanner with a CCD detector and doing Western blot analysis on this protein expression collected from the mice samples. Next, these are more examples of how they utilize this scanner to do high throughput chemiluminescence imaging. If you're interested in their publication or their research, feel free to screenshot this and check out their papers. Okay, next up, fluorescence imaging. Fluorescence imaging, I'll say, is a highlight over the few years. You can see over here, it allows you to do multiplex fluorescence, Western blotting, and high throughput as well. So they can able to do all the Western blot analysis on reconstructed human skin. And this is another publication on fluorescence imaging on a page labeling DNA on a gel. So it's not only limited to blot. You can do it on a gel imaging as well. All right, so this is a typical example of fluorescence imaging on Western blot. You have two different markers labeled at different secondary fluorescence secondary antibodies, and you can see the overlay of them. Just examine the expression levels of it. This is very good. This is another fluorescence imaging on the northern blot analysis on RNA samples. All right, so this is an in-cell Western using plate. So it will be a plate-based fluorescence imaging. You can see here these are HeLa cells are serially diluted and seeded into a 96 well and cultured and fixed, then probed for the respected targets, and then also a nuclear stain as a normalization control. Researchers have been using this laser scanner to do high resolution fluorescence microarray. You can see over here, this is basically what they use to scan fluorescence image and then invert it to black and white for better quantitation. And of course, let's not forget about IHC. So this is something that we all need it for to scan because it scans fast and then you do not need to take picture one by one. This is a motor neuron at 10 micron resolutions, brain slices, and also embryos. Sapphire can also do phosphor imaging on these carbon-14 radio-labeled plates. These images are at 50 and 20 and 10 micron resolution. You can see the details of the sample. And we have also publication that researchers use the scanner, particularly on phosphor imaging, to detect the radiolabeled samples in MSAR. This is another example of how they utilize a phosphor imaging on MSAR. They use it in an in vitro RNA decay assay. They do state over here in their own publication, say this Sapphire biomolecular imager provides sufficient sensitivity to observe all required bands with one exposure. First, it speed up their research workflow and then giving them the performance and the resolution they want. All right, protein arrays. This is an analysis of secretomes using a protein arrays from human neural discovery arrays and angiogenesis array. This is quite interesting because it's actually capture chemiluminescence signals. They use this and analyze to quantify relative quantification of all the markers needed, such as BDNF and so on and so forth. Here, lateral flow immunoassay, something that is very relevant to us because this is a novel and rapid detection for COVID-19 spike 1 antigen using human angiotensin converting enzyme 2. What they do is they develop a kit to detect COVID virus. They use the Sapphire laser scanner to do a visible and colorimetric scanning where they use to plot the histogram peaks and then analyze the data from there. They also confirm the validity of this kit using clinical samples and validate the clinical samples with the laser scanner as well. This is a very interesting paper. I highly recommend researchers to find out and look at what is happening and then how they develop a kit in this sense. Next, also lateral flow immunoassay. This time, this is used to image the 5' fam specific LAMP product on the gel. They incorporate the instrument into their LFA development for HPV-16. This is also another very interesting publication. Let's move another level up. Tissue imaging. These are mouse livers. I can see here they use the 4 solid-state laser excitation light sources. They mentioned that the application of the 4 channels fluorescence mode at 10 micron resolutions, these are captured at 10 micron resolutions, could detect gross anatomy and morphology of liver tissue, mainly based on the tissue autofluorescence. This is how they use the scanner to couple with the rest of the research and then giving you a visible image on these autofluorescence and liver. Researchers have been using the scanner to do tissue imaging and the purpose of lymphatic antigen tracking in mouse hind paw to quantify the viral infection and viral load in zebrafish, study the permeability of embryos and placenta barriers, Drosophila phenotype study, and also fluorescently labeled protein in xenopus, oocyte, and embryos. This scanner also can do whole mouse scan. You can see these are the 3 mice, wild type, transgenic, and gene set. And then they scan it on the scanner just to see fluorescent signals, whether you can detect it on the surface. Then they harvest the brain, put it on a slice, and then do a scan on it. You can see that it gives you a general idea about where is the distribution of the biomarker they are looking for. And of course, iDisco cleared mouse skull. This instrument also is capable for destacking and animated GIF generation, which meant for 3D reconstruction and switch between channels for multiplex image. This is very good for presentation wise, where you want to show people where are these markers localized at which area and then at which cells. Okay, here is a larger image showing you the merge and overlay. All of these are available in our application notes where if you're interested, please download from our official website what are the researchers have been using this instrument for with publication data and citation as well. It allows you to do chemiluminescence imaging, fluorescence imaging, and also phosphor imaging on samples that can fit up to 2.5 cm. So it redefining more in this sense, giving you the flexibility to mix and match and also do imaging in a huge batch and increase, speed up your workflow. So these are the models if you're interested in Sapphire biomolecular imager, and we have optional modules to couple with this model to fit your research need. Thank you for your time today with us. If you have any inquiries, feel free to contact us via the email shown on the screen, and we will follow up from there.